Hello, and welcome to our series on pathogen-specific considerations for investigating foodborne illness outbreaks. In this video, we will discuss specific considerations for investigating outbreaks involving hepatitis A virus. First, let's go over some of the basics of viruses. A virus is a type of microorganism that can only infect and grow inside their specific host. Outside of these living hosts, viruses will eventually die off. For example, some viruses, such as hepatitis A virus, can specifically infect and cause disease in humans. Outside of the human host, hepatitis A virus can only die off. However, because of its structure, this die-off can take a long time. Viruses are generally categorized as enveloped or non-enveloped based on their structural features. Some examples of enveloped viruses include influenza A virus and SARS-CoV-2, and some examples of non-enveloped viruses include hepatitis A virus and norovirus. Both enveloped and non-enveloped viruses have receptors that are needed in order to infect host cells. The main difference here is that the receptors on enveloped viruses are embedded in a weak lipid envelope, whereas the receptors on non-enveloped viruses are embedded in the strong capsid layer. Due to the presence of the strong capsid layer, most non-enveloped viruses, including hepatitis A virus, are resistant to many common sanitizing chemicals used on the market. For example, quaternary ammonium compounds, which is a common ingredient in many sanitizing wipe products, and alcohol, which is a common ingredient in most hand sanitizers, are not effective against hepatitis A virus. However, it is important to note that some chemicals, such as chlorine, are effective at removing the viruses from the surfaces, and some activities, such as washing hands with soap and water, are also effective at removing hepatitis A virus from your hands. Additionally, the strong capsid layer can help hepatitis A virus survive other environmental stressors and persist on food-associated surfaces for long periods of time, sometimes upwards of several months. Therefore, it is important to ensure that the right chemicals are used for various activities, such as using soap and water to wash hands and using chlorine to wash food handling equipment in order to reduce risk of transmission of hepatitis A virus. Hepatitis A virus causes approximately 10,000 cases of foodborne illness in the U.S. each year. Common symptoms of hepatitis A illness include fever, nausea, vomiting, diarrhea, malaise, and jaundice. Unlike many other foodborne pathogens, hepatitis A virus has a long incubation period that typically ranges between 15 to 50 days. Symptoms of hepatitis A illness typically last less than 2 months, although symptoms lasting up to 6 months have been reported in a handful of cases. There is a vaccine for hepatitis A, and the CDC recommends that children between the ages of 1 to 18, as well as persons who are at increased risk for infection or complications from hepatitis A, should receive this vaccine. Common local establishments where hepatitis A outbreaks can occur include restaurants and other eateries, long-term care facilities, schools, cruise ships, and penitentiary facilities. When investigating hepatitis A outbreaks linked to local establishments, there are a few key areas where environmental health investigators should aim to focus their efforts. These include evaluating whether infected food handlers may have been the source of the outbreak, evaluating if cross-contamination of hepatitis A virus from infected food handlers to food products could have contributed to the outbreak, and evaluating whether contaminated food products or ingredients may have been the source of the outbreak. First, let's discuss how to evaluate whether infected food handlers may have been the source of the outbreak. To evaluate whether infected food handlers could be the source of an outbreak, environmental health investigators should first identify whether any food handlers or other employees at the establishment showed symptoms of foodborne illness. This can be done by reviewing relevant documents, either during or prior to site visits, such as 1. Any available employee call-out records, 2. Sick leave documents, as well as 3. Other relevant documentation on recent illness reported by employees of the establishment. In addition to reviewing records, environmental health investigators should also conduct interviews with both upper management and food handlers within the establishment during investigational site visits. Some examples of important questions to ask in these interviews include 1. If and when any food handlers or other employees started showing symptoms of foodborne illness. 2. Whether there is an issue with short staffing or high rates of turnover. And 3. How sick leave policies are implemented and enforced. Gathering information on how sick leave policies are regularly enforced is especially important because if written sick leave policies are not appropriately enforced, the establishment could be at an increased risk for hepatitis A outbreaks. If one or more cases of foodborne illness are identified among food handlers or other employees based on record review and interviews, it is important to identify the exact dates in which these individuals reported showing symptoms, when they attended work, and how these dates correlate with estimated exposure dates for people affected by the outbreak. This information can help investigators identify whether the infected food handlers could potentially have been associated with the outbreak. For instance, in this scenario, epidemic curve of a hepatitis A outbreak, food handler B reported showing symptoms of nausea and malaise on July 9th and stopped attending work until after August 30th. Given that hepatitis A virus has a long incubation period, which is, on average, 28 days, mapping when food handler B started showing symptoms of nausea and malaise and when they stopped attending work to this scenario epidemic curve provides evidence to support that this food handler may have been a potential source of this hepatitis A outbreak. Additionally, in the same scenario, we see that food handler B sought further medical treatment and was confirmed to be infected with hepatitis A on July 30th. Given the long incubation period and severity of disease outcomes associated with hepatitis A acquired illness, in instances like this, where hepatitis A diagnosis is confirmed in a food handler, additional actions such as submitting press releases advising potentially exposed individuals to seek preventative immunizations like the hepatitis A vaccine or immune globulin may be warranted. For example, in a 2015 hepatitis A outbreak associated with a restaurant in New York State, after it was confirmed that an employee with hepatitis A attended work at the restaurant while they were infectious, the New York State Department of Health released a statement advising individuals who consumed food or drink at the restaurant on specific dates to seek preventative immunizations against hepatitis A virus. Next, we will discuss how to evaluate if cross-contamination of hepatitis A virus from infected food handlers to food products could have contributed to the outbreak. It is important to note that hepatitis A virus can be spread to food products from infected food handlers if they aren't frequently washing their hands or wearing gloves. And, in particular, foods that are handled after cooking or require a lot of manual preparation, such as chopped salads, deli sandwiches, or fruit platters, are at high risk for this type of cross-contamination. To identify whether food handling practices could have facilitated cross-contamination of hepatitis A from infected food handlers to food products, environmental health investigators should evaluate the practices used to prepare food products in the establishments during site visits. This can be done through observing how food is routinely handled in the establishment. During these observations, one key thing to take note of is the accessibility of hand washing sinks and gloves in the establishment to promote frequent hand washing. Are hand washing sinks properly stocked with soap and paper towels to promote proper hand washing? Are trash bins strategically placed near hand washing sinks? Is the temperature of the water in hand washing sinks appropriate to achieve effective hand washing? Are gloves available and easily accessible to food handlers? Additionally, attention should be paid to the behavior of employees with respect to their hand hygiene and glove changing practices. For example, how frequently are employees washing their hands or changing their gloves? Are employees washing their hands after using restrooms? Are food handlers recontaminating their hands and gloves after hand washing by touching their face and other parts of their body? Are food handlers wearing gloves when handling higher risk foods, such as those that are not cooked prior to consumption? Additionally, investigators can also conduct interviews with both upper management and food handlers about these routine food handling practices. Important questions to ask during these interviews include 1. How is hand washing enforced in the establishment? As well as 2. Are food handlers encouraged to wash their hands or change gloves between food handling activities? Are there instances in which food handlers are advised against using hand-washing sinks or changing gloves due to scarcity of supplies? These questions are all particularly important to ask, as any practices that deter food handlers or other employees from washing their hands or changing their gloves can potentially contribute to the spread of hepatitis A virus from infected food handlers to food products. Next, we will discuss how to evaluate whether contaminated products or ingredients may have been the source of the outbreak. In certain instances, food ingredients may be contaminated with hepatitis A virus before they are purchased or obtained by an establishment. In particular, when investigating an outbreak associated with a single or multiple local establishments, these contaminated ingredients are often sourced from local farms or small-scale local producers. Examples of some of these food products or ingredients that might be at high risk for contamination with hepatitis A virus before they arrive at the establishment include oysters or other local shellfish products, as well as various types of fresh produce, including berries or other fruit, vegetables, and herbs. To assess whether contaminated food products or ingredients may have been the source of the outbreak, environmental health investigators may want to collect select food product or ingredient samples from the establishment and test them for the presence of hepatitis A virus. Some key things to consider when collecting food product samples include making sure that sterile or sanitized tools or utensils are used to collect samples. Additionally, it is important to ensure that all collected samples are transported to the lab appropriately. More specifically, samples should be transported under similar temperature conditions in which they were collected. For example, if samples were collected at 40 degrees Fahrenheit, then they should be transported to the lab at or around 40 degrees Fahrenheit. Additionally, environmental health investigators should be sure to communicate key details about sample collection with the lab before collecting food product samples, including the quantity of food products to be tested and the organisms that they should be tested for. This is particularly important for hepatitis A virus, as not all labs will have a protocol in place or the equipment needed to perform testing for hepatitis A virus in food products. In summary, in this presentation, we have covered 3 key areas that may be relevant for environmental health investigators to consider when investigating hepatitis A outbreaks linked to local establishments. In addition to hepatitis A virus, check out our other videos in this series that cover key considerations for investigating foodborne illness outbreaks linked to other foodborne pathogens.